Hello, good morning, you all. I'm Andrea Cuoghi, working for Bonfiglioli Engineering. I'm a technical space specialist, and in this presentation, as well, will help Gianmarco Pincelli, his technical sales manager of the same company. So we start with this presentation. It's about the quality inspection methods, and especially we are referring to Pharmacopeia Annex 1 that is focused on sterile product manufacturing to allow worldwide exportation of pharmaceutical products. So we will focus at the first topic on the leak detection methods. So first basic question, what CCIT stands for? So CCIT is an acronym and it stands for Container Closure Integrity Test. So basically this is the leak test. Leak tests are very important to detect different kind of problems on the packaging of a product, and of course it's really important because it's in primary, it's in contact with the final product. Being a product, a drug, is very important that the packaging should be preserved. So problem and defects on the packaging, defects causes different problems. So for example, loss of product that can flow outside. Critical headspace gases could be contaminated or go outside of the product, and that's very bad because it's there exactly to protect the environment. And other big problem is contamination of microorganisms from outside. This, of course, have a very bad impact on the drug because it's not anymore effective or can even have a bad effect on the final customer. So this, of course, have to be kept under control. Let's have an overview of the market of CCIT worldwide. Last year, we were speaking about 1.2 billion, but the forecast for less than 10 years is to more than double this value. So we can see from here how the market is growing. So this means how important this is for the manufacturer that are always more and more having this kind of controls and technology in their plants. We can see from the graph that everywhere this control is foreseen to grow. North America, Europe, Asia Pacific, Middle East, and Latin America. So everywhere this is growing. It's not just focused in some part of the world. Going more in detail, the not destructive methods are growing because these are much more reliable, they are deterministic, and of course they don't affect the production, so it allows to earn, to save money. More and more there are used control on 100% of the applications, and this is helped as well because the technology and the inspection machine are now easier to be found in the market, and this of course is a big improve that help the process in a plant. Important is to remind that FDA estimates that more than 40% of the recalls of the product is due to packaging failure. So almost the half of the problem on the market could be due to packaging failure. And from here we understand how is important, how we should really implement this kind of control in our facilities. There are some main guidelines and laws that are regulating the market and the control that have to be done for guaranteeing the quality we want to have. So the most important reference are the European Pharmacopeia that are very important guidelines that say when and what we have to do as inspection. The USP, the United States Pharmacopeia, in 1207 chapter speaks in detail about the different technique, the different methods which we can use to make this control and this analysis. And as well, the FDA is caring about and speaking about in its content. Then we have, going more in detail in Annex 1, we can see that they are manufacturing practice, but we have to follow them. They are general rules that we should follow to guarantee the needed quality. So, as we said before, Annex 1 does not mention any specific validated method. For the validated method, we should jump to the US Pharmacopeia. But the indication, very important, that this Annex 1 gives us is that for closed by fusion product should be subjected to 100% integrity test using validated methods. This is very important because this forces us to make this kind of control. For larger volume, a reduced sampling may be acceptable, but anyway, it's strongly recommended as well for the bigger ones to proceed to 100% integrity test because of the guarantees this gives to the quality of our process. Samples, products that are using system different from fusion, other than fusion, should be anyway be inspected. And even if not 100%, it should be justified why the sampling is less than 100%. And the recommendation is always to measure, to check 100% of the production. And this is very important for the production and as well for auditor. The auditors that are making their check, their inspection, if we can prove, we can show that we are inspected as 100%, for sure is added value for our production, and it's everything smoother. The second main rule that we are going to inspect is the 1207. Then is going more in detail in the different methods. So it applies to vials or bottles, form, fill, seal plastic or glass ampoules, syringe or cartridge, flexible bags or pouches, and packages for some drug device combination. It speaks more in detail about the test method, limits of detectability, basically the leak size range, and it splits among deterministic methods and probabilistic methods. And of course, deterministic that are more repeatable. Of course, the accent is very positive about these methods, while the probabilistic is less. We can be less confined on this kind of methods. That's why it's stressed how important it is to try to follow deterministic methods. Let's jump inside this split, as I said, between deterministic and probabilistic methods. Between the deterministic, we have pressure decay and vacuum decay. These are like parallel technology, we can say. Laser-based headspace gas analysis, mass extraction, tracer gas, electrical conductivity. While on the other side of the table, so probabilistic methods, we have microbial challenge by immersion, bubble test, liquid tracer, sniffer mode gas tracing. The most of the probabilistic tests are destructive. Of course, this is a disadvantage of this kind of methods, and often they need a big preparation that must be done in a very accurate way, and it can depends on different operators that are making this preparation. This means repeatability depends as well on the human part, while a machine is much more repeatable. Deterministic methods are more, we can be more, it's more reliable, let's say. Most deterministic tests are not destructive. Of course, this is as well a very positive point for deterministic methods. And in the last few years, we have seen a transition exactly for the point we have discussed right now, and we will see as well later on, to the deterministic. As I said before, here another table with advantage and disadvantage. Deterministic methods are reproducible, are sensitive, highly depends on instruments and or physical laws. Quantitative test result outcome, minimal test sample preparation, and so the risk of error is very low. While on the other side, the probabilistic methods are not reproducible, are not so sensitive, that don't depend much on instrumentation, are more qualitative, interpretative results. Considerable test sample preparation, this implies a lot of time lost, and the quality, how is prepared the sample, is affecting the final results. So again, it depends a lot on the human factor. And at the end, the risk of error is high. That's why the rules, the norms are always stressing the importance of follow deterministic methods and possibly 100% of the sample to be inspected. So the publication of the new USP guidance demonstrate a clear preference for deterministic and not destructive methods. So now I leave to speak my colleague Gianmarco, because we are now jumping more in detail inside the different technology that we have mentioned up to this moment. So please, Gianmarco, if you want to follow.
Thank you, Andrea. We discussed about which are the methods listed in USP 1207. USP 1207 is just touching the methods, but is not touching the other thing that should be considered in order to decide which is the best method to leak test a certain product. So I listed here some important points to be discussed, which is the qualification of the instrument. So we have to take care about to understand the qualification of the equipment used for making that test. Easier qualification is less work to do, is easier. So as well the method development and validation. So as well the validation package is a validation package that is compliant to USP 1207, is referring to that norm, is strong enough to be presented to some auditors eventually. So we have to make this question yourself in order to decide which is the best one. As well, listed in USP 1207, there are some information about to generate some negatives and positives samples, okay? So no leak samples and with controlled leak samples in order to understand if the technology can be applied in your product with the real leakaging. So fill it with the final product inside with a hole, eventually laser certified in flow instead of diameter is really important, using the final package. So this is really important. As well, we have to understand that some semi-automatic equipment needs the operator that is loading and unloading, so we have to take care about as well to understand the costs of using person if the machine is not fully automated. As well, we have to take care about the CFR 21 Part 11 compliance, about the electronic signature that should be implemented in all pharmaceutical equipment for production. So take care about to verify if that is inside of the machine. What are the most common inline validated methods? So USP 1207 is listed a lot of methods that are mainly for laboratory. But in reality, if someone wants to have something for small production batch or inline, which are the methods, the common method used for that? And there are listed here. We have the vacuum decay, pressure decay, so differential pressure, positive and negative, outside of the package. We have the HVLD, so electrical conductivity, and we have the headspace gas analysis, so the system, the technology that is measuring the gas content in the airspace in order to understand if there was some leakages on the product or not. This headspace gas analysis is more, we can say, a process control, but in some cases can be applied as a CCIT. Starting from the vacuum decay method, so basically the container is inserted inside of a testing chamber, okay? Then we have inside of testing chamber our eye, that is a vacuum transducer that is monitoring constantly the vacuum level outside of the container. Then we have a solenoid valve connected to a vacuum pump. So for a certain time, this solenoid valve is opened and the vacuum pump is connected to the external environment of the product and the vacuum is generated. Usually high vacuum is generated. For liquid contents is more than the vaporization level of the water. Okay, so we can say approximately 10, 15 absolute millibar will be reached outside of the container. Then this solenoid valve is closing and the vacuum transducer is measuring the decay over the time that there is outside of the container in case of leakages. If you take care about the graph, here we have the green graph that is representing the constant behavior of the vacuum outside of the container, characteristic of conforming product. Then you have the red one that is decaying constantly because there is one airflow that is constantly coming out of the product. Okay, it's important to understand when we are thinking about differential pressure technology like vacuum decay and pressure decay. Uh, it's, it's important to see that the machine is detecting a certain leakage in terms of orifice diameter, but is more technical, saying that the machine is looking for one airflow because the vacuum is decaying due to the airflow and not due to a certain hole diameter. Okay, in an indirect way, yes, but there is a correlation in between the airflow and the hole diameter that is as well affected by the thickness of the walls on the container. So moving to the second technology that we have, so we have the pressure decay method. This pressure decay method is really effective with solids products that can clog the leakage if the leakage is in front of the product. For example, oily product content in the ampoules, like that one that we showed here. If you have the hole in front of the oil cannot be taken out from the product through a really thin path because the viscosity of the oil itself is blocking. So, in case of positive overpressure outside of the container, so the system is exactly identical to the vacuum decay, but instead to have the vacuum pump, we have the compressed air. So we generate approximately outside of the product one positive bar gas, okay, outside of the product, and the air can enter through a thin path. So you see some bubbles over the test that are entering. So exactly the same. So we open the valve for a certain time, we reach a certain pressure level. If the pressure level is maintained constantly over the time, nothing is entering on the product, so the product is conforming, no leakage. If you have a drop in the pressure, means that the air is getting inside of the product. So typically this is application for oilies, I said, as I mentioned, as well as suspension, powder, lyophilized product, because yeah, every time there is drying the product, so the process is clogging holes there. So it's important to have a positive pressure outside of the container. So what about the validation method? So differential pressure are mainly managed by this ASTM F23809 that is as well recalled in USB 1207. So this is really important because explains very well how to apply that to a leak testing system based in differential pressure. So basically can be applied to all type of containers, so ophthalmic dropper, glass or plastic ampoules, vial with elastomer, lidded, threaded bottle. So is applicable for all type of container, as well IV bag or pouches. Okay, so it says don't check the behavior of the system, so the capability of the system to detect the leakage on the container, making a real container on the real holes, real leakage on the product, because is too variable the result that you will get after making real holes on the problem of the product. Sorry. So the result of the leak test, if you make some laser drill hole on the product, is really different because we have some factors that are changing the airflow that is passing through that device, that the product drilled. Okay, so it's saying using a calibrated leaker a calibrated leak outside of the container. So if you remember, when we were talking about a vacuum decay method, I said that was better to focus our idea not to a whole diameter, but in airflow. So for example, if you're talking about 10 microns, the correspondent airflow is 0.87 cubic centimeter for a minute. So these norms is saying put this 0.87 cubic centimeter for a minute, if you're talking about to identify 10 micron salt in a device, and activate this device inside of the system. Don't make real holes in the product. And the approach is exactly that one. So, for example, in case of vacuum decay, we add that device called calibrated leaker, okay, is certified in airflow. So through that device are passing just 0.87 cubic centimeter for a minute. We add another solenoid valve. What happens when we are opening this solenoid valve? The air is entering from the atmosphere through that device and is decaying the vacuum level outside of the product exactly in the same way that will be from a real leakage on the product. So the effect in the measuring, what is measured by the transducer and the machine is the same. So this is really easy because that is really easy to be validated. You have a certified leakers, you can activate it, you can demonstrate to the auditor that this calibrated leaker is certified. You can as well measure the airflow in front of them if they want. But in this case, the validation of the method and the qualification of the method is really, really strong. And is not affected by the container, by the thickness of the wall, for example. So, discussing about the vacuum decay method, some pro and some cons, is perfect for water-based container. As I said, the level outside of the container of the vacuum could be really high, and you can reach the vaporization of the water. That is increasing the capability to detect a really, really small leakage. Is emphasized the leakage detected if some water is coming out from the product and evaporates. The evaporation of a certain quantity of liquid is 10 times more than the same quantity of air that is coming out of the product in case of what is measured by the machine. So the vacuum decay is 10 times more if you have the vaporization. So this is really important because for water-based product, this is a really high level of test. So you can detect a really small leakage like 5 microns, 10 microns along these numbers. So as I said before, is really to be validated and qualified because you have the STM that is suggesting you to do that, and you can do that on the machine. You can imagine that if you have a rotary machine with 60 heads, you have 60 calibrated leakers installed in all heads that can be really easy activated over the production. So you load on the container, you load conforming container of the machine, we switch on the function of the machine, and you are activating all the calibrated leakers in all heads. So you are challenging not one head, you are challenging all the heads of the machine has, because that should be two. Then it's not affected by the filling level. So you can have a different filling level on the container and you don't have any type of different behavior in the result of the machine. You need to have some air head space. This is really important. About the cons, you need for inline, you need a rotary machines. So there are not linear machine, are rotary machines. So you have multiple heads. So we have impact on cost. So if one is price oriented, for sure this vacuum decay is, we can say, a Ferrari, but it's costly because you have heads. Okay. It's not applicable for clogging products. So if you have oil-based life product, is not applicable. Machines are not so easy. So when you have multiple heads, you need to synchronize all heads with the handling system. And you need to have a really high experience supplier. So don't take this machine from a new company that is making from this machine from 1 year or 2 years. Okay, you need to have this machine working for 20 years. Okay, so focus on the experienced suppliers. A problem of that, a cons of that, the last one, sorry, Andrea, I come back a little bit. The last point, okay. So you have the head station contamination. Because when you have a gross leak on the product and you can spill, we can say, water on the product, the testing heads can be contaminated by this cross leak. And in this case, we have a special function on the machine that automatically dry the testing chamber that got wet from the previous leak test. Okay, go ahead, please. So pressure decay method is the second one, differential pressure, positive, as I said, perfect for easy container, not for, we can say, flexible container. It's highly suggested for clogging products like oily product, lyophilized suspension, and so on. Is as well validated following the STM, so is as well stronger like the other vacuum decay, and as well you need a rotary machine. So same consideration about that one, with the important consideration that overfilled product can impact the test sensitivity. So in vacuum decay, you can see, we can say, basically the evaporation of the liquid, although there is a really low quantity of air inside, in case of pressure, we have to check if that certain overfilled product can be leak tested using a pressure decay method. Then listed in that common, we have the HVLD. So HVLD, it's using a current, a microcurrent that is passing through the container. So this is listed in USP 1207, is really huge applicated to all type of products, so plastic product, glass product, but we have to make some consideration about that. So first of all is that the current is passing through your container, and you have to make all the considerations about that. Because in case of pinhole, the current that is passing through the container is different. So the container is working like we can say isolator, and the liquid inside of the product is working a conductor. So if you have the leakage, is generated one, we can say, current path inside of the container instead of outside. So it's taking the short part, the short street, the current. So you have a different behavior, different absorption of the current from the product. So the product is less isolated if there is a pinhole. Then you can reject the product that has one low absorption of the current. So this is basically the physics of the technology. Then what about the validation? So we say that for pressure decay and vacuum decay, we have the STM that is supporting us. This is not true for the HVLD. So HVLD has not rule that allows to validate the machine with something we can say artificial generated unless there are real holes on the product. So in order to qualify and validate the HVLD, you need to have this laser drail container. So we have to have positive. We have to generate positive product in order to understand if the system can detect that for the qualification, but as well we have to make the day challenge. So in the opening of the batch and in the end of the batch, we have to pass on the machine this laser drilled container, so positives container, in order to understand if the machine is still able to detect what should be detected in terms of a limit of detection. And this is really important because if you don't have nothing as an international standard that allows to use other system, you have to buy, we have to store, and we have to fill this type of positives with the final product as well inside. So it's a system that has to be managed with the real defective sample. That is not so easy, everything on that. There are some suppliers that can provide some, we can say, artificial generated product like a dummy, with a resistance inside in order to simulate the passage of the current inside, taking the short path in case of leakages. But as well, this is not listed in any type of international standard. It's just a other system, but is really difficult to defend something different than laser drill hole, real hole in front of the auditors that are asking about that product. Okay. So electrical conductivity as a pro, for sure, is a cost-effective solution because the machine is inline machine. You can have different heads to leak test the product because normally the part that is tested by the HVLD is just a part of the container. So you are not testing the whole container like in pressure decay and vacuum decay that is inserted to a testing chamber. So we are testing the whole container. In this case, HVLD is testing just a part of the container. So the lower part, for example, then you have to rotate the product, move the product, twist the product, then you can test the other part. So we have different heads in the inline machine, usually are inline machine. So we have different electrode points, 3 or 4, in order to inspect the whole product. We can say that as a pro, in some area of the product, the HVLD is really sensitive. So you can detect really small holes in a certain point, in a small area of the container. For example, if you have one ampoule and you would like to focus the high sensitivity of the machine on the tip because it's the welded part, is the tip, is good. You can do that. Be aware that you can detect a really small leakages dust on the tip because if you move farthest you are moving from the tip, lower will be the sensitivity. So the sensitivity is the system is dependently of where the hole is on the product. As a cons, yeah, we have a lot of cons because yeah, it's using the conductivity. So it's not applicable for product that are not conductivity or have a less conductivity than 1.2 microsiemens. For example, oily, powder, lyophilized product, suspension, obviously, that cannot be inspected using HVLD. Just some part of the product can be inspected, and another important thing is that the current passage that is passing through the container can kill some drug composition. For example, if you have live cells, we make one test, for example, with a customer, and we saw that HVLD with their product killed 25% of the live cells. It was a PET therapy injections. So in this case, HVLD is degrading the product. It's degrading for the current, but it can degrade the product as well for ozone creation. Since that some lighting is stacking outside of the product and inside of the product, you can have ozone creation. The ozone creation outside can be managed with the vacuum system to protect the operator for the inhalation. Okay, but inside of the product, if the product is closed and you have some stack of light inside of the product, it's really difficult to detect it. So you put on the market product with ozone inside. Yeah. Then, for example, if you have some plastic container like blow-fill seal, in some points this plastic is born to be melted easily. The HVLD can melt this type of plastic in a certain point of the container where the thickness of the plastic is not so huge. Okay, in the corner, for example, in that point, if the light stuck here, you can make some porosity on the product. Then, if you have some empty container, because a big leakage, so you have a big leakage on the product and over the production chain you are losing all the content, that cannot be detected by the HVLD system because the product is empty. So if you don't have any conductor inside of the product, the current is not taking the shortcut and is not detected. Okay. The last one listed in the four technology is the headspace gas analysis. So headspace gas analysis is based in TDLAS, so tunable diode laser absorption spectroscopy. Is listed as a CCIT, as a not destructive method. But we can say that it's a technology that can be applied as a CCAT just in some special cases, not in all cases. You cannot see I need a space gas analysis system to make the CCAT on my product. You have to make some investigation if the space gas analysis is applicable to your product and if can be used as a CCAT method, because not in all cases can be used. There are a few examples later on. So how it works, first of all. Okay, so it's based on near infrared diode, that light that is passing through the container. So you need to have one emitter. We have one emitter and one receiver. So the light is emitted and is received. If you have some absorption of the light from the oxygen molecule, for example, you have a different light that is passing through the container. So for example, if I set the laser wavelength to the oxygen level, to monitor the oxygen level, that is 780 nanometers of wavelength, okay, that is absorbed, okay, by the product. In this case, the product, inside of the product, you have oxygen that is filtering the light that is passing through. That means that you have oxygen. But you have to understand if this oxygen can change over the time due to leakage on the product. So you need to have a differential pressure in this case in between the outer air and the inner air. So what you can measure with the headspace gas analysis, basically for the CCIT, you can measure these three things. You can measure the oxygen percentage with a wavelength of 1854 nanometers. Okay, this is more a process control than a CCIT, measuring the oxygen inside of the product to control the nitrogen flow, for example, on the capping machine. Then you have the relative humidity measurement. In this case, as well, you are measuring the humidity inside of the powder contents in order to avoid the conglomerate. Third application on that is the total pressure. So we use the same laser used for the humidity. Then in a special point of reading, when there is the absorption of the water, we can measure directly the total pressure inside of the container. And this is typically applied as a CCAT, for example, for lyo product. So for lyo product, you have the vacuum inside, and the laser has to measure a certain level of vacuum inside of the container. So few millibars, for example. If you have one hole and you need a certain quarantine, and this is really important because longer is the quarantine in between you cap the product and you inspect the product, smaller will be the hole that the machine, the leakage that the machine can detect, the system can detect. So you can imagine you cap the product on the lyophilizer machine, on the lyophilization system, okay? You cap the product, you wait 5 minutes, and you make the measurement. You are expecting to have few millibars. If you have 100 millibar, 200 millibars, means that something was entering on the product, but you don't know if it was a real leakaging or we can say was a capping defect on the machine. You are not able to do that. You are not measuring the leakaging of the product like on pressure decay and vacuum decay. You are measuring just the effect of a potential leakage on the product. Okay. Validation of a space gas analysis system is used making some standards. For example, if you have oxygen, so you purchase, we purchase tanks with a certification of the oxygen percentage, we cap the product and we make that standards. These standards as a controlled level of oxygen inside. So we put on the machine, for example, the 0.5%, the 4% and 10%, and we check the result of the machine. As well for the humidity as well, we use the chemistry, so we use some saturated cell solution in order to generate the standards with a certain controlled level of humidity inside. About the application of phase gas analysis in the USP 1207, it's really important to understand that is the effect on the time. So you need to have what I call it quarantine. This is an example of a test we made. We take one 8R tubular vial, we make 5 microns all on that, and we pass the product on the machine several times from 17 minutes. Okay, so you can see that in this vial we generated 3 absolute millibar inside of the vial. Okay, you can see the match with the standard. So we made, first of all, 5 standards, one at 3 millibar, one at 50 millibar, one 100 millibar, 200 millibar, and 400 millibar. And this vial was passed on the machine to see the result. Okay, you can see the unit. For example, 100 millibar gave out the result of 90. Is not unit of measuring. Is 90 is just a number, a poor number. Okay. So you can see that in time zero, you have the graph, the readings. These are really multiple reading over the time. You can see that the pressure is constantly raising over the time. After 2 minutes, you reach inside of the vial 50 millibars, starting from 3. After 4 minutes, you reach 100 millibar. That's it. This is an example that we got 400 millibar after 17 minutes. In this case, having the vacuum inside of the container, although there is normal oxygen percentage like 20.8, we are not measuring here the oxygen, we are measuring the total pressure inside of the container. In this case, we detected 400 millibar in 17 minutes. And this is application for vacuum measurement inside of the product. Another example, this one is for oxygen. So the vial in this case was filled with nitrogen inside, but with 800 absolute millibar inside. So minus 200 millibar goes inside of the product. So slightly vacuum and nitrogen flow to protect the product. As you can see, starting from the point zero as well here, in this case, we are measuring the oxygen. So oxygen should be absent inside of the product, but if you have a leakaging, some air, atmospheric air with oxygen is entering on the product, and as you can see, after 15 minutes, we can say we had a really good separation, okay, on that, and after 40 minutes approximately, we get 4% of oxygen, because as well here we made standards. The violet line was 4% of oxygen. So in the beginning we had 0%, we have 5 microns sol, the air was entering on the product because we have delta pressure, delta vacuum of 200 millibars. Then this vacuum pull inside the air and the oxygen was growing. So in this case, we are using oxygen measurement to make a CCIT system. If you don't have this differential pressure in between inside and outside the product, we can say the applicability of using space gas analysis SSAT is not so strong. So we have to make some other thing. But my suggestion is apply this space gas analysis SSAT just in case of differential pressure in between inside and outside your product.
Okay, so I take another time the speech. We are now finished the part of CCIT, and we face another very important part of the inspection to be done on this kind of packaging and drugs, and this is the visual inspection. Let's start again from the rules, from the norms that are regulating the standards. And we start from the United, as before, with the US Pharmacopeia, European Pharmacopeia, and as well Japanese Pharmacopeia. In different chapter, they speaks about the visible particulates in injections and visual inspection of injection. Sorry. And all these three norms speaks about, and what they say is basically is the same. So they speaks and says that all the filled containers, for example, this is an extract from Annex 1, all the filled containers of parenteral products should be, should be inspected individually for extraneous contamination of other defects. So it stresses the importance, and it's really a must to check each product to see if there is any contamination, any particles inside. The batches that have different levels from the standard of particles or some criticalities inside must be stop the production and make an inspection, make an analysis about what's happened and why there is this situation out of the standard. A defect library should be generated and maintained where there are stored all the class of defects. That should be generated as well in consideration of manufacturer experience and based on risk analysis. So in this way we can have different class of defects, like critics, relevant, or simply aesthetic problems. We will see it later on in the presentation. Annex 1 from European Pharmacopeia and USP 1790 say the same about the quality, let's say the expectative of the result of these inspections. And they say that the inspection methods must have a result equal to or even better than manual inspection. So this is very important chapter because it says what should be the performance of an inspection method. So it has always to be compared by human visual inspection. A lot of different questions can arise, exactly because what I said before. For example, what is the minimum particle a method, a machine, a device can detect? It depends on a lot of factors. It depends, for example, on viscosity of the product, because maybe spinning the product, the particle can move or can be stuck on the weight of the particle, on the color, because it's easier or difficult to be seen. It depends on the surface of the packaging, because if it's plastic, and it's not transparent, could be much more complicated than if it's glass vial, for example. So a lot of parameters affect the result of this inspection. But what really important is the comparison with the human. So which is the particle size that the machine can detect is not the relevant. What is relevant is that the machine must have a equal or better results than human inspection. And this is stated in US Pharmacopeia 1790. Okay, to make this comparison, it must be created, as we said before, a defect library, where inside there are different kind of contaminations. So this could be generated during production, because the visual inspection made by human can detect and isolate this problem, creating so a library. Or the other way that a manufacturer can follow is to ask to some supplier. I mean, there are companies that are able to generate this defects library based on the experience and the information given by the manufacturer, that is supposed to make a risk analysis and give this information to the supplier, that will be then able to generate this library. Then qualify operators have to make a check of this and evaluate in this way the probability of detection. That will be the challenge for the machine. This library is called the KNAP kit, and we will see later on, Gianmarco will introduce this, this new chapter, speaking about how it's composed, how it has been done, and how to challenge the machine. So I leave you the word, Gianmarco.
Thank you, Andrea. Basically, the NAP test kit should be generated following the USP 1719 by multiple operators and multiple times. Okay, just to as well answer some questions that arise on the chat. Okay, so this NAP test kit is exactly saying which is the percentage of detection of each container that is composing the NAP test kit, and as well which is the false rejection rate by the human, made by the human. Okay, because inside of that should be composed by 250 products. You have as well conforming product that operators are saying as a non-conforming. So as well the human is making the false rejection rate. Obviously cannot be compared by the machine, but neither the speed can be compared the human and the machine. You cannot compare the speed. So it's a compromise. The false rejection rate should be considered in any case when you are purchasing a visual inspection machine automated. So the rules is to use 3 or 5 operators, 5 seconds in the white background, 5 seconds in the dark background, then take the decision. Not 6, not 10, just 5 seconds. Otherwise, the NAP test kit is not done as should be. Ok, vai por avanti, Andrea. So the NAP test kit should be composed by 3 defects: critical, major, and minor. Critical defects are the ones that are compromising the sterility of the product, okay? Like particulate inside, as well leakages, but leakages are not inside of the NAP test for visual. You should use a different method, like a CCIT with calibrated leaker. In this case, we are thinking about extrinsic, intrinsic, and inherent particulate inside. Then you have the major defects that are not compromising the sterility of the product, but that are compromising the usability. You can imagine, here I make the example of a blow-fill seal. If you have the hook is missing, you cannot hinge the product injecting to the patient. Or if you don't have the hook to open it, you cannot open it. Then we have minor defects that are basically cosmetic defects, inclusion, black spot, stretches on the product. So normally what we suggest is to use 170 conforming product and 80 with defects. So the kit should be composed by 250 products. So about the detectability of the particulate. Okay, there are customers that are asking me which is the particulate that your machine can detect. As Andrea mentioned, it's not a question that we can answer because depends of a lot of things. Anyhow, some studies was done from different scientists like Knapp, Borchet, Melchior. So a lot of data we have here, and we can state, and it's for sure, that in a transparent tubular glass container, 10R, if operator takes 5 seconds in the white and 5 seconds in the black, the 70% of the detectability is reached about at 140 microns of black particle inside of the product. In average, okay? So doesn't matter which is the minimum particulate that the automated machine can reject, because should be equal or better than a human. This is an example. This is an example for this type of container. And just to give you an idea about how difficult is inspecting the product, I will introduce here the word DIP. That means difficult to inspect products. Okay. So starting from the right side, from the left side, sorry, we have the transparent container with a transparent liquid inside. Easy, we can say. False rejection is low. We are outside of the DIP. Okay, is really applicable. Then you are moved to a difficult container. You take the same container, amber. Okay, is a little bit higher the difficult, a little bit higher the false ejection rate. Then go ahead like that. So we have the transparent molded container that is forced, the amber molded container, worse and worse, moving in the plastic container like LVP, SVP, blow-fill seal container, or transparent container with solid. As you can see, the inspection of powder and LIO is really difficult because the false rejection rate is high because you cannot detect everything. First of all, you cannot detect everything that can be detected by the human because the human has more experience. We have some AI application on that mitigates that, but this is just to giving you some idea about how difficult inspecting some types of product. In the right, there is the white column that is exceeding the practically not possible. Just to remind you that maybe some product cannot be automated inspection. I didn't feel that chart because for the moment we don't have any application that is practically not possible, but it exists. Earlier or later, you will find something that cannot be inspected. So this is just to wrap up about the difficult to inspect this type of product. Okay, move ahead. Yeah, so algorithms can detect really easy bubbles in this case to mitigate the false rejection rate. In this case, our AI can detect a bubble having information about the position and as well where is the black part. For example, in this bottle, if you can see in the plastic container, the bubble is stuck on the walls in the left part of the container, and the black part is in the left. Probably this bottle managed by the algorithm was in the lower part, so the dark is low. So AI is learning about that and is mitigating the false rejection for this type of product. Okay, move ahead. Okay, as well for product like that, that is a blow-fill seal bottle where the bubbles are stuck on the walls as well. Here, algorithm can detect this bubble as a bubble and is not making any cosmetic defect in this case. So a normal system is identify this bubble as a reject. System based on AI can learn about the bubble position, bubble characteristic, and can avoid to make false rejection for that. So last slide is about the container closure integrity. So we say that should be done 100%. In some cases, we are still using some CCIT based in statistical. Remember that the quality is really important for this product. You can imagine that some product can arrive on the market contaminated or not effective. Uh, I mean, product that was degraded by some leakages and they are not getting injected to the patient to cure the ill, maybe the cancer. Okay, you are expecting to have something that is working but is not working because it's contaminated. So this is really important, and our driving force is suggest to be safe in the patient side and apply 100%. Although the USP 1207 is saying that you can use SPC.
Thank
Thank
you
you so
very much,
much.
Gianmarco and Andrea. We're actually out of time, but there was one follow-up question from Gilad about very hard to compare to human visual inspection, false positive and false negative. How can we know?
Uh, exactly in the same way I said during the presentation. So on the NAP test kit, you have all the defect container, so container with the particulate inside that was rejected by the operator a certain POD. So you have a container that is rejecting 86% of the time from a container from the human inspection, a container that is rejecting 90%. When you pass the NAP test on the machine, you have exactly the same behavior. So the machine is giving you the result, okay, this container was rejected 87, this was rejected 95. So you compare you can compare number with number, really easy. As well, the force rejection rate, you have the operator force rejection rate, as well you have the machine force rejection rate. So you can really easily compare, it's not very hard. If you make a proper NAP test kit and you pass a NAP test kit to the operator, I should do, and in the machine multiple times, you have a really chart where you have manual inspection result, machine inspection result.
Okay, great. Thanks, Gianmarco. So we
You're
are
welcome.
actually out of time, but if anyone has any questions and they would like to pose them to the speakers, please do get in touch with us. We will then forward that on. As mentioned, this session has been recorded and it's saved on the ISPE Singapore YouTube page so that you can go back to that and review. We'd like to thank our speakers, Gianmarco and Andrea, for sharing their experience with us this afternoon, and also to everyone for joining us. Until our next session, we would like to wish everyone a great evening, great day, and we want to thank Bonfiglioli for sponsoring this session. So thank you, everybody, and it's been great to hear from you all. And until our next session, take care. Thank you, gentlemen, and good evening to everyone.
Thanks. Bye. Good evening.
Thank you, everyone. Bye
Bye
bye.
bye. Thank you so
Bye
much.
bye. Thank you, everyone.